Connect the device to the computer using a USB cable. Plug in the device's power supply and turn on the power switch. The smart breathing light will illuminate. Open the software and check the connection status in the bottom left corner. Once the connection is successful, a status indicator will appear in the bottom right corner. Click on Experimental Operations to access 4 modules: Gel Experiment, Luminescence Experiment, Fluorescence Experiment, and In Vivo Experiment. Open the door and place the black background board into the lower slot until it touches the switch. Place the sample on the background board, and then proceed with imaging. Luminescence imaging can be conducted using the upper or lower field of view. Select the Luminescence Experiment module. Bright field imaging is recommended, and you can enable the auto option. The module provides options for single-shot imaging, continuous imaging, and cumulative imaging. Single-shot imaging. First, demonstrate single-shot imaging. Set the exposure time or choose auto exposure, depending on the experimental requirements for high sensitivity or high resolution. Select different pixel binning values. A higher binning value results in shorter exposure times but blurrier images. Conversely, lower binning values lead to longer exposure times, but produce sharper images. Before capturing the image, click Preview for real-time visualization. Confirm the preview image is good, then proceed with imaging. After imaging is complete and the image appears, review the results. You can adjust the upper and lower CTS value limits on the right to achieve the desired imaging effect. Optionally, select the invert color function based on your needs. Continuous imaging. You can capture up to 10 continuous images. Adjusting the first exposure time value will automatically update subsequent times. Cumulative imaging. Capture up to 20 images in cumulative mode. Open the door and pull out the transillumination light box. Press the switch button located on the right side of the light box. Place the sample on the glass panel of the transillumination light box. Push the light box back into the device and close the door. In the software, select the Gel Experiment module and click Preview. You can choose between the transillumination light source or the illumination light source. Set the exposure time manually or select auto exposure. Once configured, click Capture to take an image. Manual gel cutting. If you need to manually cut the gel, open the door and pull out the light box. Place the sample on the glass panel of the transillumination platform. Insert the orange gel cutting protective shield into the slot. And turn on the excitation light source to begin cutting. If you need to image protein gels, place the white light conversion plate onto the transillumination light box. Then place the sample on the white light conversion plate. You can then proceed with imaging. Place the background board in the lower slot and position the sample on the background board. Select the Fluorescence Experiment module in the software. Choose the desired fluorescence channels. Set the exposure time or opt for automatic exposure. You can select up to 5 light sources simultaneously. Click Capture to begin imaging. Place the background board in the lower slot and position the sample on the board. Select the In Vivo Experiment module. Choose between luminescence imaging or fluorescence imaging. Luminescence imaging. Set the exposure time or click Auto for automatic exposure. Adjust the pixel binning value as needed. Fluorescence imaging. Select different excitation and emission wavelengths based on your requirements. Once all settings are configured, click Capture to begin imaging. After imaging is complete, the captured image can be used for data analysis. There are 2 types of ROIs analysis for in vivo experiments: manual analysis and automatic analysis. Manual analysis allows you to select the desired area as needed. Click on the analysis results, and then export the document. Automatic analysis can perform automatic recognition, and then analyze the results. Automatically generate an Excel spreadsheet when exporting. Multi-image analysis allows for analyzing the same region across multiple images. Begin by selecting the region of interest (ROI) on the first image. Click one-click copy to replicate the selected ROI. Choose the images you want to analyze and click Confirm. This feature enables comparative analysis of the same region across different images. Click 3D Dynamic Scan to generate 3D image. You can have a more intuitive view of the signal strength. There are many pseudo-color types to choose from. Click Save Image to save the required 3D image. Click Reset the original image to restore the modified image. You can choose to export a single image, multiple images, or all images. There are 3 export methods. Export for publishing. Exports images in TIFF format for gel experiments, only the imaging map is exported. For luminescence experiments, bright field images, luminescence images, and overlay images are exported. Export for analysis. Exports images in raw.tiff format. Export BLT images. Exports in BLT2 format, including raw data and TIFF images. Multi-image combination export is also available, allowing multiple images to be exported as a set. Image information. Click on the image information to view image information, such as experiment ID, experiment time, image type, pixel merging, exposure time, and light source. Click on the operation log. Click on the operation log, operation status, and operation time. Includes image settings, lighting settings, device information, language settings, etc.